So we have an extremely distinguished keynote speaker to kick off this conference. It's Professor Glenda Gray from South Africa. And I have to say that doing a bit of homework on her last night, she is the most distinguished speaker I think that I've ever introduced. So we're very, very lucky to have her. Her list of achievements is pretty long, so this is just some edited highlights. Professor Gray is president of the Medical Research Council of South Africa and professor of pediatrics in the Faculty of Health Sciences at the University of the Witwatersrand. She's a pediatrician and a scientist. She specializes in the care of children and in HIV medicine. For her significant contributions in the field of prevention of mother-to-child transmission of HIV, she was awarded South Africa's highest honor, the Order of Mapungubwe, I hope I pronounced that right, as well as the Nelson Mandela Health and Human Rights Award. She is co-PI of the HIV Vaccine Trials Network, which is a transnational collaboration for the development of HIV AIDS prevention vaccines, so lots of clinical trial design experience and conduct experience. So Glenda, I'd like to welcome you to the podium. Thank you so much for coming all this way to talk to us. The stairs are over there. Yep. And I'll let her introduce her talk. Glenda, over to you.
I wasn't sure if I was able to see, but anyway, I've got my glasses here just in case. So I'm going to speak today about HIV prevention trials in the area of pre-exposure prophylaxis and its impact on trial designs and the ethics of standard of care in low and middle income countries. So it sounds like a mouthful, but the kind of complex trial designs you'll be thinking of, we and the ethics of standard of care is something that we in the HIV prevention field are living with at the moment. And so I just wanted to give some background to why I'm giving this talk, is that the standard of care for treatment of chronic diseases and the prevention of diseases for clinical trials in low middle income countries, although governed by various ethical principles which you are quite well aware of, require more precise articulation when it comes to its execution when it's not in the comparator arm. And so the first question in South Africa, when we were considering the issue of pre-exposure prophylaxis, is who is responsible to provide it? The sponsors don't, if you're doing public-funded studies from the public purse, it's not normally the responsibility of the institution to provide standard of care. It's the responsibility of the government. And when the government is not providing the standard of care and the sponsor can't, what do you do in those situations? Who should provide it and by whom? So we also had the discussion about should it be provided in the context of the clinical trial or should it be referred out? When you're doing a phase 3 vaccine trial, people are very busy, and do you want to divert them by getting them to provide standard of care, and can you come to some kind of relationship? So some of our ethics committees believe it's the clinical trialist's responsibility to provide the standard of care, and that creates issues of equity and perverse incentives. Will people come into your trial because you offer PrEP? And how do you distinguish about people coming for a vaccine trial as to people coming for accessing standard of care? And so those are some of the issues that we spoke about. I've mentioned the issue that sometimes standard of care is simple if industry is running the research. But when it is coming from the public purse, the issue of who is responsible for standard of care becomes a big problem. There's the issues of access after the trial. So although we're doing HIV vaccine trials, if we offer PrEP in our vaccine trial and the trial ends, and there's no PrEP in the public sector, what is the obligation of the trialist in that situation? We also have issues of acceptability, feasibility, how implementable and cost-effective are these interventions. And so, as you know, WHO had clinical guidelines to to suggest that countries should provide PrEP for people who were at risk of HIV, who were at reasonable risk for HIV, and they set the incidence around 2%. So if you were living in a country that has an HIV incidence of more than 2%, the country should be providing PrEP for those populations. What WHO did not take into account was the implementability, the feasibility, the acceptability, and the cost. Countries are very poor. South Africa has the highest HIV burden in the world. We have 7 million people who are HIV infected, and the government fiscus spends most of its time trying to afford a treatment program. And asking the country to divert to providing PrEP meant that there was a costing issue, and PrEP was not a best buy in South Africa. And so South Africa is a sovereign country. There's a WHO clinical guideline. There's a recommendation, and they sit between a rock and a hard place about expecting the public purse to provide PrEP. So those are the kind of things that we had to think about. There are trial design issues when it comes to PrEP, and luckily, unlike Ebola and Zika, the RCT trials, the clinical trial design, are probably much easier to implement and to think about than when you're dealing with a complex epidemic like Ebola or Zika. So we are sitting in low middle income countries, and HIV prevention is critical. And we have high incidences of HIV, and if we don't do studies in countries like South Africa, we will never solve the problem of HIV. And so unfortunately, the burden of disease means that the burden of the trials will occur in low and middle income country settings. So with HIV, we have 4 prevention opportunities. We have opportunities that'll enable the unexposed person to protect themselves, and that's like using STDs, circumcision, treating STDs, circumcision, and condoms. If you are exposed, this is the space where I work in. You can look at interventions like vaccines, pre-exposure prophylaxis, microposides, and neutralizing antibodies. And then if you have been exposed, there are the issues where vaccines may still work, and post-exposure prophylaxis may work as well. And then in terms of infection, treatment as prevention will make people who are HIV infected less viremic and therefore less likely to transmit HIV onward. And so most of the work that we are doing in South Africa is in the area of preventing HIV acquisition after being exposed to HIV. So these are the current scalable biomedical interventions that you could consider when you were doing your trial design. And one of the most important things at a community level is treatment as prevention. And so one of the best ways to reduce population incidence is to make sure that everybody who's HIV infected receives an early diagnosis, goes immediately onto treatment, and prevents ongoing transmission. And that's one of the ways that has been touted to try and end the HIV epidemic. Medical male circumcision has been shown to be consistently effective in reducing acquisition. And we have medical male circumcision programs that are rolled out. And when we enroll men into our trials, we refer them for medical male circumcision. And depending on the site, there will be variable uptake of medical male circumcision. Now PrEP has been consistent. Has been consistently effective in discordant couples and in men who have sex with men and intravenous drug users. However, PrEP in women has been much more variable, and you can see the various confidence intervals for FemPrep voice and the voice overall, and the voice that looked at the tenofovir oral medication. And so you can see when you're thinking about an intervention or standard of care in women, it becomes much more difficult to understand what's happening and what kind of message do you give women who enroll in your trial who need to access PrEP, and what do you say to them about how efficacious PrEP is? Because overall, in heterosexual women in Southern Africa, we have not established efficacy or effectiveness. In various subgroups of women, we've established effectiveness, and there's a correlation between adherence and efficacy, but also there are a lot of biological interactions that may render the intervention not effective. And so if you give women PrEP in a prevention trial, the message must be, there's very important messages, is that PrEP probably in women is unforgiving. And so if you're not adherent and you're not taking your PrEP 6 to 7 days a week, this is not going to work for you. So don't give up on your condoms. And so women need to know that PrEP may not be the silver bullet that'll prevent HIV acquisition. And if we're not careful about the way we message PrEP in prevention trials, we may mislead women into thinking that they have a highly effective intervention and they don't have to use condoms anymore. And so these are the kind of things that we grapple with when we talk about PrEP in treatment trials for women. So today, I think it's the right time, and we've been thinking about trial designs and how to design trials in HIV, given the interventions that are rolling out. And it's the right time now to think about how we're going to design the next generation HIV prevention trial in order to make sure that we can have a license, we can get licensure for the HIV prevention modality that we use, and that the trials that we run are feasible and scientifically valid. I'm sure you might be aware of the cost of HIV vaccine trials. So the HV10702, which the trial I'm the protocol chair, which we're busy executing in South Africa, costs $135 million. And so that's a lot of money. And so as you get more and more complex, the price of a trial goes up and up and up. We estimate that a single arm in a vaccine trial costs around $50 million. And so every time you add on an arm, it becomes up. And we always expect the NIH and the US taxpayers to fit the bill for these kind of vaccine trials. And just recently do we have public-private partnerships, and only recently do we have pharma willing to invest as well. And up until now, it's been a public-private partnership with philanthropy like the Gates Foundation, the NIH, and just recently companies like Janssen agreeing to foot some of the bill for these vaccine trials. And so in low and middle, I mentioned a little bit about low and middle income country PrEP access. And it's been incredibly complicated. So although we have this WHO guidance that says PrEP should be given in countries with high HIV incidence, the access has been limited by the limited access to PrEP in the public sector. So if you're in South Africa, the only way you can get a drug in the public sector is for your drug to be on the essential medicine list. And so, so what happens is that there's a committee that sits and considers very carefully each drug that it has to put on the essential medicine list, because you have to consider the cost and the priorities. And so, so we expect the committee to sit next year to think about whether PrEP should be on our essential medicine list. And there are lots of principles for medicine selection. You can see that it has to have a public health relevance. So does PrEP have public health relevance? Is it safe? Is it effective? And how do we deal with the issues of women? Can we afford it? And what are the implications for practice? And so these are the kind of things that we consider. And we also consider how robust the information is to get onto our essential medicine list. And so we look at all the data and we look at the evidence. And so when you look at alternative trial designs, maybe think of yourself as the guy on the committee in a country that has to look at your data and make a call on how your evidence looks in order for it to get onto the essential medicine list. And so obviously for us, the level 1 to get onto essential medicine list is a lot of RCTs that keep on confirming the data, and obviously a high quality individual RCT, which is 90% power, will obviously be the golden standard to get onto our essential medicine list. But this is an important thing as you design trials to try and think about how do you convince a poor country that the data that you're presenting is good enough for the public purse to pay for it and to put it into the essential medicines list in your country. So these are the kind of things that we look at. Because of these, the variability of access to PrEP in our country and the issue of standard of care, we in South Africa had a discussion. We had a standard of care summit, which was hosted by the South African Medical Research Council, to try and understand the kind of standard of care issues and debates that we have and what we should be doing. We were in a situation in South Africa that although we're rolling out a whole lot of HIV prevention RCTs, we had some ethics committees that started to withhold or put trials on hold because they were saying that we need better strategy on how to access PrEP in these trials. So we decided to host a summit to talk about standard of care in low and middle income countries. And what we came out with from this summit is that the standard of care considerations are incredibly complex and nuanced in the developing world, particularly when your drug is not on the essential medicine list and not available in the public sector. What do you do then? For us in South Africa, PrEP is an aspirational standard of care. So although there are country guidelines and there's WHO guidelines, less than 7,000 South Africans are accessing PrEP. And so what do you do when a standard of care is only aspirational in your country? There's also the debate about who should deliver PrEP. Now, ethics committees are saying that the clinical trialist in the clinic is responsible for making sure participants get PrEP. And some ethics committees only believe that's the way it should be. Other ethics committees are saying to us, you can get a partner in the community, you can speak to a public sector partner, you can look at a demonstration site and you can refer your participants into them. We also have a debate about who should get it in our clinical trials. And so we enroll not only young women, we enroll men and we enroll older women. Now, if you look at our country guidelines, the country guidelines talk about equity, but they do target specific populations, and those are young women and men who have sex with men. So if you are a man in our vaccine trial, or if you're an older woman in our vaccine trial, what do we do and how should we deal with that? Because that goes above the standard of care in your country, but yet it's important in terms of equity. Again, we had the responsibility of who was it to deliver the PrEP, and then there was the issue of equity versus perverse incentives. And so what do we do in a situation where PrEP is not available in your community and people come to your trial because they want PrEP? And should we be giving PrEP to the community in a clinical trial? On how do we manage that? So it's very complex, the issue of standard of care, and not as simple as one would think it would be, and requires a lot of further clarification. So we're working now with our communities and our clinical trialists and our ethics committee to try and construct a way to deliver PrEP in a way that is equitable, yet doesn't put the onus of the PrEP provision on the clinician who's in the study, whose job it is to deliver high quality data so we can license a biomedical intervention. So what are our considerations now in terms of HIV vaccine trials? So for non-HIV vaccine trials, I think it's very easy. HIV PrEP, for non-HIV vaccine trials, PrEP has become the comparator arm. But as we get better and better PrEP options, we are going to require correlates for HIV incidence as it becomes more effective, or in populations where PrEP efficacy is high, like in MSM or in IDUs. And we require more innovative markers to see what the HIV incidence is in those communities. And obviously things like STI and behavioral risk scores have been touted. And so currently in our HIV vaccine trials, we're collecting over time behavioral data to develop scores and STI data. We're actively screening for STI so that when we do our future trials, we have some markers some indicators or correlates of HIV incidence that will help us develop the trial design and understand the efficacy of future vaccines. So we're really thinking about that for our next trial designs. The observed efficacy in PrEP in women is variable, as I've shown you before. And so when we do look at this in a comparative arm, in the comparator arm, it's very easy to see why it's a superiority design and not a non-inferiority design, as we've seen in men. And obviously, as PrEP becomes more available, we will see that it'll be in the background as a background standard of care, as seen in like condoms and STI treatment and PEP that we give out in trials. For HIV vaccine trials, having correlates of protection or correlates of risk will enable us to design future vaccine trials for bridging. So once we understand the mechanism of HIV protection, it's going to be very easy to quickly bridge to other populations or design iterative studies, because we won't have to do large-scale phase 3 trials anymore. And for future trials using PrEP as a comparator arm, we would look at a counterfactual placebo group. I think they will be discussing this in the future. And a counterfactual placebo group will be used to estimate the effectiveness of each arm. So what we envisaged in the future is probably doing a 3-arm study, looking at vaccine plus PrEP, or vaccine and the counterfactual placebo and PrEP and the counterfactual placebo to try and estimate the individual effectiveness of each arm. And those are the kind of studies we would probably design going forward. So if you look at our current repertoire of HIV prevention trials in South Africa or in Southern Africa, we have the ECHO study, which is the hormonal contraception and HIV risk. We have optimizing PrEP strategies. We have novel design for microbicide effectiveness. And we have the next generation PrEP trials looking at long-acting intramuscular antiretrovirals, and then we have the HIV vaccine strategies. But just quickly on the hormonal contraception and HIV, so there is a possible risk, a possible HIV acquisition risk with some of the hormonal contraceptions, and there is an uncertainty in the data. And we know that there are life-saving benefits of hormonal contraception. So we have equipoise here. We have a contraception that may cause HIV risk, but without that contraception, you may get pregnant, and it may impact on your mortality. And so obviously, this is a public health conundrum in the developing world. And so the ECHO has been designed to look exactly at that. So 7,800 women have already been enrolled between the ages of 16 and 35 into DMPA, into an implant, and into the copper IUD, and are followed over time. So at this moment in time, there is no PrEP access in ECHO. And post-summit, we have set up a fund that women sites can apply for funding to purchase PrEP. So women in this study can also have access as background standard of care to PrEP. We have the 028 study design, and this basically looks at how do we optimize, given the variability of PrEP efficacy in various populations, how do we optimize PrEP? And this 082 is a very nice study because it basically looks at people, if they want to accept PrEP, they get randomized to provide PrEP with an enhanced support versus just standard of care. And those who decline, they receive the standard of care and are offered PrEP if later on they want to go and get PrEP. And then once they get PrEP, they would be then randomized to receive PrEP with the standard of care adherence versus the optimized standard of care. And so basically, the intervention looks at the current adherence support and then looks at enhanced adherence support. And so the whole objective is to try and optimize PrEP adherence so that we can get better efficacy. And the important thing about this is to look at the proportion and the demographics of women who accept or decline PrEP, to try and understand what are the issues there, and then to try and improve PrEP adherence, looking at drug levels in women. So that's a nice, interesting study design, and allows women to decline, but it still allows women to be followed up in the study. The Depiverin ring is a microbicide study. There were two components to the Depiverin ring study, and this was a phase 3 study that looked at a microbicide ring with an antiretroviral in, and looked at this was in Southern Africa, and basically followed up a woman to see what the outcome of this was. And as you can see, this ring study was moderately efficacious, if you, between 27 to 31%, and in the ring study as compared to the Aspire, and also variable. I've lost one slide. Doesn't matter. Sorry. Okay, and then just basically variable efficacy according to age, which complicates the microbicide study even further, because not only do we have modest efficacy, depending on the age that you would have even more variable efficacy. So the lowest efficacy was in women under 21, and the highest efficacy was in women over 25. And that's probably a combination of adherence. Maybe women who were under 21 were taking out the ring, and women over 25 were less at high risk and more likely to keep the ring inside. And so that is complicated. But post-trial, there is a rollover, looking at an open label rollover to look further at long-term safety and adherence. And this open label study enrolls 2,000 women, some from the ring, from previous ring studies, and another 300 ring-naive women, 18 to 21, to try and understand the issues of efficacy in that group, and then older women as well. And basically, women are randomized either to receive it on a monthly basis or to receive it on 3 monthly basis, where they're given the ring to take home. And this is a nice study to try and evaluate further the safety and efficacy of it in a less stringent design, but hopefully will give us some evidence, some insights on incidence and how effective it is. HP1003 is a long-acting cabotegravir study. So this is the next generation of oral PrEP, and this study is designed to look at the effectiveness of this in men who have sex with men and transgendered women, and essentially is comparing standard of care with cabotegravir. And this is a non-inferiority study, and it's important to understand that, because there's enough evidence of efficacy of PrEP in MSM and transgendered women to design a non-inferiority study. Whereas in women, this is a superiority study, because the FDA could not determine the point efficacy of oral PrEP in women. And so the design is completely different, because we don't know how effective PrEP is in women. And hopefully this study will show us again, will further articulate exactly how effective oral PrEP is in women. But these studies are starting. In fact, they were screening some of the participants last week in South Africa. And so these studies will enroll over the next 4 years. In terms of HIV vaccines, I'm going to go quickly over this. We've got 3 strategies already in the field. We're looking at a clade C approach to preventing HIV vaccine in a phase 2b/3 design. We're also looking at a multi-clade approach, and the first proof of concept, HVTN 705, has just started last week. We enrolled 10 participants, and then we have our neutralizing antibody approach that's trying to test whether neutralizing antibodies can prevent HIV acquisition. And so 702 was built from the famous RB144 TAR study. As you can remember, there was declining effectiveness of RB144 over time, and that at 1 year post-vaccination, the effectiveness of the efficacy of the vaccine was 60%, and it declined rapidly to 31%. And so the South African trial has built on RB144. We've made a clade C vector. We brought a new protein in, and we're doing boosting to try and keep up the durability. And we really tested the TAR study in South Africa to make sure that the vaccine was immunogenic in South Africans, And we've constructed the Clade C program. HV10100 is complete. We met the go, no-go criteria, and we started HV10702 last year, and we already enrolled over 2,000 South Africans in the last year. And this study will show us whether we can build on RV144. It'll show us how potent the vaccine is, and also how durable it is by building in additional boosts. What's also important about 702, we'll be well powered to look at correlates of protection. And this is very important as we design our next vaccine trials, to understand what are the biological correlates and what are the genetic correlates, and what are the host and virological issues that impact on vaccines, so that we can build the next vaccine trial and also probably make vaccine design much easier. The multi-clade approach is a collaboration with Janssen, and we're looking at a vaccine that will protect everybody against HIV, not just in the Clade C epidemic. And this is a global vaccine approach using an Ad26 prime and a trimeric boost. And this study has a precursor to it called HV10705, which has just started in South Africa, and is based on the non-human primate data that showed using an Ad prime and an an Ad26 prime and an Ad26 prime boost with an envelope protected in non-human primates. And the correlates of protection seem to be ELISpot and ADCC. So using both human trials in the TIE study and in using non-human primate data in this study, we can hopefully build on our correlates to help us design our future HIV vaccine trials better. And this just shows you the sample size and the efficacy. So for 702, this is a true hybrid phase 2b3, where we're enrolling 5,400 participants, mostly women, and we've designed the study to have a power to be able to reject the null of less than 25%. And in 705, this is a proof of concept, and we've designed it's a phase 2b study, and we have designed it to have a 90% power to reject a vaccine efficacy of less than 0. And these two studies have been designed in such a way that we're breeding participants for peak immunogenicity at the same time points to be able to look across those two areas. I'm not going to go too much into this. This just shows you how important correlate studies are, because it helps us understand the causal mechanisms, both the basic science and how to manipulate the data we see in non-human primates, and trying to understand the passive immunization. It's also important for statistical associations, because it helps us assess efficacy. It helps us assess what were the correlates of vaccine efficacy. And this is just a diagram to show what we look at when we're looking at correlates. So the importance of correlates is helping us to shorten trials. Trials are 36 months long, and reducing the cost. That's a very important thing. It also helps us to guide future vaccine development, and also to bridge between basic and clinical science. It helps us guide regulatory decisions and helps us guide immunization policies, and also hopefully helps us bridge vaccine efficacy to new settings, which we need to do. We can't continue to do these huge trials. We need much more elegant ways of trying to develop efficacy and to bridge to other studies. My last few slides is just looking at the neutralizing antibody approach that we are doing in HIV vaccine research. And as you know, there's an arms race between the virus and the antibody from the time of transmission. And the virus replicates rapidly all the time, and the immune system never ever seems to catch up. In a few participants who before ARVs were widely available, a few participants developed broadly neutralizing antibodies. Given new technologies, we've been able to clone these and make neutralizing antibodies, monoclonal antibodies, to infuse into people to see whether they can protect against HIV acquisition. And these studies will test for the first time whether we need neutralization to protect against HIV acquisition. The first of these antibodies to go into clinical trial is the VRC01 study, which is an antibody against the CD4 binding site, and that's enrolling in 2 studies, one in MSM in North and South America, and one in sub-Saharan women. And this is a 3-arm study, and this is important to try and understand which dose we need. So this is a 10 milligram versus 30 milligram versus placebo, and this will tell us the minimum amount of neutralizing we would need, and will help us understand issues around scalability. So obviously for us, the best one is to have a low dose, the low dose work, because then it becomes easy to make it into a subcutaneous injection rather than an infusion. These studies are almost enrolled, so we hope to finish the enrollment in Southern Africa at the end of January this year. So how do we design, after I've showed you all of these trials, how do we design HIV prevention trials in 2021? So in 2021, we're going to have all this wonderful data from the long-acting antiretrovirals to the neutralizing antibody studies. We're already designing triple monoclonal antibody studies. How are we going to design the studies in 2021? So what are we going to do? What should we be thinking of? And how do we take these things to scale? So I think that our interventions should be tested in the field for population impact and for evaluating scale-up. So if we have moderately efficacious biomedical interventions, we need to put those in the field. And we need to look at population impact, and we need to see how we scale these up. Looking at the kind of designs, probably a step wedge design may be the most optimal, but community randomized trials may be important if we have different interventions that are not going to be used together. We may see a community randomized trial where you would look at long-acting antiretrovirals versus vaccines. And so those are the kind of studies that we could be thinking of, which will require much more complex designs and much more nuanced endpoint evaluation, both in the primary and secondary. And we also need trials to elucidate the optimal combinations of vaccines and non-vaccine interventions. And this is where the counterfactual placebo group is very important, because we're going to have to see how all of these interventions work together. And so I've given you a lot of information and some of the kind of dilemmas that we face, some of the ethical dilemmas in terms of standard of care in South Africa as we try and unravel this. So we haven't solved the solution of standard of care, and I'm also giving you a taste of what's coming down the pike and how we balance the issues of finances together with optimal trial design, and at the same time making sure that we can scale up and deliver these interventions to the communities that need them. Thanks very much.
That was a fabulous talk. Thank you so much, Glenda. Glenda has very, very kindly agreed to take some questions, I believe. I hope that's right. Any questions on that amazing overview of HIV trials and the complexities of doing them? I don't know. We'll put this in the...
talk in terms of, thank you, in terms of the PrEP and the comparator arms, especially given the considerations of maybe PrEP being delivered by the health infrastructure already embedded in the community, what are the issues with ramping up task coverage as well in the communities, given that treatment as prevention has far higher demonstrated efficacy in terms of thinking about sort of that as being the standard of care in the communities in which you're conducting the vaccine trials?
So you said task shifting. Did you say task shifting?
Sorry, treatment as prevention.
Yeah.
So ramping up
okay.
testing.
Yeah. So so my country, so South Africa, largely funds its own treatment program with the help of the Global Fund and PEPFAR. And every time a new intervention or a new guideline comes out, we do cost effectiveness evaluation because it's coming from the public purse, and we choose what we call best buys. You know, what are the best buys for our money? and where we're going to get the biggest bang for our buck. And so our government did the sums and has concluded that the best buy is treatment as prevention, and to scale up treatment and diagnose, to scale up diagnosis. So we're introducing self-testing, community-based testing, trying to triage people into care, and trying to impact on community incidents. If you have a look at our data in South Africa, although we've increased life expectancy by 9 years since rolling out antiretroviral treatment, we haven't impacted yet on new infections. And why haven't we impacted on new infections? Because most of the infections happen very early on when you're asymptomatic. So half of the infections, half of transmission occurs in the first 6 months after HIV acquisition. And so if you can't identify new and acute infections, you're not going to stop HIV acquisition. So treatment as prevention is important, but we're getting prevalent people onto HIV treatment and not the people that have just seroconverted or the acute infection. And so we need we need better testing so that we can get the people that really need to be on treatment to prevent HIV transmission. And so that's our biggest issue. The country is experimenting with demonstration projects as far as PrEP is concerned, and is very cautious about how to scale it up because it's costly for us. As you roll out, if you can imagine, 20 million people would require oral PrEP, and this would be forever. And so the intervention gets more and more expensive as you roll out, whereas treatment as prevention gets cheaper because we have a finite number of people who are HIV infected. If we can get to that 7 million people, then we can probably contain the cost and use treatment as prevention as the best strategy to contain community viral load. If that answers your question.
Time for one more question, if there is one.
So, hi, I'm Eileen Dunn. I'm here in Bangkok at the Centers for Disease Control. I had a question about a term you used called counterfactual placebo or counterfactual in your clinical design alternatives. Could you explain that a bit? I've never heard that term.
Okay, now you're putting me on the spot here, as I'm a clinician. So counterfactual placebo group. So when you don't have a placebo arm, and you have interventions that you're evaluating, you want to see the strength of that intervention by itself. And so the way you would design a study would be a 3-arm study. So I'll use vaccines as an example. Vaccine plus PrEP in one arm. You randomize to vaccine and PrEP in one arm. Then you randomize to vaccine plus a counterfactual placebo. Placebo arms, and then you randomize to the PrEP plus the counterfactual placebo group. So you have a 3-arm study, and so you would then have the power to look at how group 1 is, how the effectiveness in group 1 versus group 2 versus group 3, and then you can do some. We're the statisticians in the audience. So from what I hear from Peter Gilbert, who's my statistician at Sharp, is that you can then look at the individual effectiveness of each of the interventions because you have this placebo group in the second and third arm, and you'll be able to start to discern the individual effectiveness as if it had a placebo arm. That's the way I understand it. Maybe the statisticians in the group, if I've explained it wrong, could clarify. But as a clinician, that's how I've understood his reasoning in terms of that. And I think that's probably the way we would go in HIV vaccine research, is looking at that. There are other ways. There's David Dunn. They talk about so the issue around looking at how do we know what HIV incidence is in a study without a placebo arm, and so looking at other biomarkers like STDs, we haven't been able to validate a biomarker or a correlate for HIV incidence yet. It may be STDs, it may be this behavioral risk score that I spoke about. And so we need to start collecting other adjunct data to try and understand what would be the HIV incidence in a community if you didn't have a placebo arm. And so we try to look at some of the correlates, like some of the biomarkers, like STDs. So we need to work on those kind of things. And so some of the alternative trial designs in HIV vaccine have taken into account looking at biomarkers or looking at other ways of inferring the attack rate and the reduction of infections in that arm.
Okay, thank you once again, Glenda, for a wonderful overview.
Thanks.
We could give her a big round of applause, please.